So when we are talking about polytene chromosomes, this is about the history of it, right? So we know that Balbiani was a scientist who had first observed rather, and reported these were giant chromosomes in the salivary glands. When he was observing the cells from the salivary glands, and the cells were in the interphase, right? So several DNA strands which are placed side by side, and that appeared as a very big molecule rather than the normal appearance, what it would be under light microscope. So here, these interphase chromosomes, they were commonly seen in salivary glands or diapteran organisms, right? So diapteran, yesterday we have seen a few examples of diaptera. The common one that you have been studying is the Drosophila, right? Just now, 5 minutes back, we saw a video that showed how those larvae from water were collected, how they were dissected, and the salivary glands, dissected salivary glands were stained with acetocarmine, and how the polytene chromosomes were observed. The larva of which species? Yes, the name is very much there here on the screen. CHIRONOMUS larva. Right? That means, how do you spell it? Yes, who is there on the screen? Ishan? Chironomus larva, right? Fine. These are the very larger chromosomes. They're normal, and they are easily observable under compound microscope. So here, these are the insects belonging to Diaptera group. So adult leg fly or other fruit fly, that is Drosophila, and these are the larvae of those animals, of those insects. So when you talk about these insects and their larvae, we are referring it to with respect to polytene chromosomes. Now, when it comes to morphology of polytene chromosomes, very important feature that you need to mention it, they are seen during interphase. The sizes of the giant chromosomes are with 5 long and 2 short arms. If you remember the diagram that we had seen yesterday from the book, here I'm sharing it again. So this is the larva. These are the salivary glands. From that, these polytene chromosomes are observed. And this is what is the arrangement of interband band, gray band, and dark band. So when you talk about the giant chromosomes, it has got 5 long arms and 2 short arms, which radiate from a central point, which is called as chromocenter. So the length of these chromosomes, if you see, is about 2000 microns, whereas the somatic cell size itself is 7.5. So how compactly it is arranged, that you can imagine. Now, chromosome is formed by fusion of centromeres of all the chromosomes. 2n is equal to 8, right? So 2n is diploidy, we are talking about it. So you are going to see one of these photographs that tells you about the types of the different arms radiating from the chromocenter structure. All right. So let's see enlarged image. So this is chromocenter. This is chromosome 4, X chromosome, chromosome 3. Now this is the right arm, left arm, another left arm, another right arm, right? So these structural details we are expected to show when we are going to draw the diagram representing polytene chromosome. So this is chromocenter, point number 1. Then you have chromosome 4, X chromosome, chromosome 2, chromosome 3, right? And what it is showing, normal mitotic chromosomes at the same scale. It is just for comparison, right? Now, region where the 2 chromosomes, which are homologous, are separated. Homologous, that means structurally they have same sequence, same gene, same arrangement. They are separated here. So here you see a common start and common end, but here are 2 arm like structures, as if it is a forked form at this point. But this shows what? The homologous area homologous sequences in those chromosomes, which are synapsed. Now, if you see the dimensions, it is 20 micron size, right? So if it is a single one. Now, against this scale, you can just compare the size of this giant chromosome. Now let's see in the details of these chromosomes. So when it comes to morphology, we have seen X chromosome longest arm, Y chromosome is fused with the chromocenter and is usually not seen as separate strand. Then 2R, 2L, 3R, 3L, and 4 chromosome. So all these parts were seen in the diagram above. All right. So chromatin or polytene chromosomes are staying in banded pattern. So yesterday we have talked about what these bands are, how they are categorized as gray band, dark band, and interband. So the dark band represents heterochromatin, whereas the light bands, they represent euchromatin. About 80% of the DNA is located in bands and about 15% in interbands. That means only 15% of DNA is genetically active in polytene chromosomes. Now we talked about Balbiani, who first observed and reported these polytene chromosomes, but further details were highlighted by the scientist, painter, and Bridges in 1930s. See, it's almost a century time that lapsed between the two very great observations and reports about the same structure, that is polytene chromosome. So what they observed, there are more than 5,000 bands, and yesterday we have seen how they are differentiated. 5,000 means 1,200 are dark bands, 2,400 are the interbands, the gray bands, and rest of them are the duplicates, right? So why is it so giant? So it is about 1,000 times larger than the other somatic chromosomes, and as long as 0.5 millimeters and up to 20 millimeters in diameter. So lengthwise 0.5 millimeters and diameter-wise 20 millimeters, and that's why they are looking giant. So the larger size of the chromosome is due to the presence of many longitudinal structures called chromonimata. Right? So they also are called as polytene chromosomes. That means many threaded. Poly is to many, tene is to threads or long elongated structures. So many strands of the giant chromosome are due to the repeated divisions of the chromosome without cytoplasmic division getting completed. That is separation of the replicated chromosomes by attachment to spindle fibers and separation by spindle fibers, the mitotic division ending up as telophase, and two separate cells are formed normally. But when it fails to happen so, incomplete cytoplasmic division will lead to formation of these structures, and that is nothing but resulting due to endoreplication or endoreduplication, the term is. So multiple copies of the same chromosomes, so many sets of those chromosomes will get generated. For example, a chromosome that undergoes endoreplication 10 cycles, and there is no separation of the daughter chromosomes and no completion of mitotic phase, then 10 10 times the chromosomes is replicated by 10 times, 10 cycles it has undergone. So how many copies of that you will get is about 1024 identical strands of chromatin are going to get formed, and they're going to be spreaded line by line or aligned to each other. That's why it is going to appear as a giant structure, and which is held together by chromocenter. Right? Now, in the process of making these multiple copies, very important thing is multiple copies are happening, but segregation is not taking place. So this occurs when the DNA repeatedly replicates and there is no karyokinesis, that is division of nucleus is not happening. So you are going to have amplified number of copies of chromosome. Now, what about the mRNA formation, protein synthesis in the glands then? If so many chromosomes are formed, but they are not going to be in the interband section, then what about its expression, right? So this is the banding pattern. We'll come to the expression part after going into the details of these banding patterns. The polytene chromosome contains two types of transfer bands. These are the dark bands and the interbands, right? The dark bands are darkly stained and interbands are those which are lightly stained with the nuclear stains. Dark bands are going to contain more compactly arranged compacted DNA and less RNA. But in the interbands, you are going to have more RNA and less DNA because interband is loosely compacted. It is undergoing expression. So it is the active part of chromosome, right? So the difference between bands and interbands is with reference to staining due to compaction or less compaction. And second is on the basis of how actively functioning they are. The dark band chromosome regions are non-active at the moment and the lightly stained are actively functioning. That means they are undergoing transcription and translation. Now, the bands can be recognized by their different thicknesses and spacings. Each one has been given a number to generate a polytene chromosome map. So chromosome mapping is another word that you are coming across here. In this context, we'll talk in the next lecture when we talk about use of gene transfer methods, natural gene transfer methods in bacteria and mapping of genes. In that context, we will look into this concept. Coming back to how many uh, polytene chromosomes are present that we have seen. What is its organization that we have seen? So how many base pairs per polytene chromosome? So yesterday we have talked about 30 to 60 kb with respect to Chironomus larva. Now in case of Drosophila larva, you could find 50 kb to 1 lakh base pairs. So 100 kb base pairs DNA which is present in the polytene chromosome. Shall I go to next slide? So polytene chromosomes, as we have seen, they are not going to remain silent forever, but depending upon the growth phase of that organism, the specific regions of those genes are going to be activated. It's easily understood that interband regions are under expression, but what about tightly packaged gray and dark bands? So they are going to loosen up, decompact, decondense, and those regions will appear like expansions, or they are technically referred to as puffs. The puffs appear in the polytene chromosome. Puffs are nothing but the decondensed regions. So the initially condensed part get decondensed, the inactive part becomes active, and the expression in those regions occur. So the radio tracer technique, in which labeled bases are incorporated into the RNA of these developing larvae, and then that polytene chromosome is extracted to observe what is happening actually in the puff regions. So the puff regions would demonstrate RNA synthesis because that can be easily traced out by radioactivity due to the radioactive element that got incorporated into the RNA that got synthesized in these actively expressed regions. New RNA is getting formed. So that new RNA formation will pick up new components like RNA के जो components है उसमें you are going to require carbon, nitrogen, phosphorus, etc. So these elements, if it is a de novo synthesis that is starting from each element builds up that nucleotide and from that nucleotide to polynucleotide, that is RNA, then this radioactively labeled RNA will be observed and will be formed, and we can observe that radioactivity, that is the emission of beta particles that can be tracked using the tracer instruments. All right. So it will tell you about yes, RNA is getting synthesized. That means those genes are getting transcribed. So activity has begun. All right. Now, when we come to chromosomal puffs or They are also referred to as Balbiani rings because the scientist who referred to these polytene chromosome for the first time in 19th century, 1800. So the bands of polytene chromosome get enlarged. To these, we refer to them as puff, and the process is called puffing, where the expression of those genes is occurring. The regions of puff, the chromanemata uncoil and open out to form mini loops. Thus, puffing is caused by the uncoiling of individual chromomere in a band.